All right, welcome back to the MCAT prep series, and today we're going to be continuing the biochem section by talking more about amino acids and specifically peptide bonds, disulfide bonds, and hydrolysis reactions. So, just to cover the learning objectives pretty quickly, by the end of this video, you should be able to understand peptide bond formation. You should be able to distinguish between peptides, polypeptides, and proteins. You should be able to understand disulfide bond formation, and finally, be able to explain hydrolysis of peptide bonds. Okay, so moving into the content, just a quick review from last lecture. Here is our basic structure of an amino acid. So as you can see, right in the middle here, we have a alpha carbon connected to a hydrogen and an R group. And just a brief reminder that the R group is unique to each individual amino acid and sort of determines its structure and its functional properties. And then more importantly for this video, we're going to be focusing on the amino group and the carboxyl group because these are the two primary groups involved or responsible for the formation of peptide bonds. So moving on to today's lecture, what actually is a peptide bond? Well, a peptide bond is the bond made between two individual amino acids in order to create one peptide. And so a peptide bond is an amide linkage. And so the way that peptide bonds form is you have an amino group, a free amino group from one amino acid, which attacks the carboxyl carbon and another amino acid. This results in a loss of water. So H2O is lost as a result of this reaction. And then the two amino acids that were individually separate combine with each other. So I'm just going to draw out what that might look like. All right. And then so one thing you might be asking yourself, just after quickly looking at this reaction, is where this water or where this H2O even came from. How is it possible that H2O is just produced? Well, as you can see here, in the final product, you just have this carboxyl oxygen and then one hydrogen attached to the former amino group. So that means two of these hydrogens and this oxygen are not in the final product. And that's because they're actually what composes that new water molecule once this peptide bond has been made. And then one additional side note is just that this reaction or this nucleophilic attack right here is called a nucleophilic acyl substitution. And one good thing to be made aware of is that this new peptide that is formed has what we call an N-terminus, which is around the free amino group, and a C-terminus, which is around the free carboxyl group. And so typically when we're writing these peptides, we go from N-terminus to C-terminus. That's the direction that they're traditionally written in, and that's also the direction that they will be typically read in. Okay, so moving on, let's take a look at this peptide bond, in itself. So one thing to note is, like I said before, this bond or this peptide bond is also called an amide bond. And that's because it's between this former carboxyl group and this former amino group. So this is the amide bond. Okay, and one thing to note about this bond is that it does exhibit partial double bond character, which means there are resonance structures related to this bond. And as a result, it does have a relatively rigid structure and restricted rotation about it. So whereas, you will have rotation around other bonds in a peptide, you can always assume that this bond is relatively rigid. Okay, so let's look at that, those resonance structures. So as we know, this nitrogen has some free electrons around it. So it can occur that these electrons move down here, and then this double bond moves up here. And so the resonance structure you're now going to have around the peptide bond is an oxygen with a negative charge because it has three pairs of electrons around it, and a double bond right there. And so now, if you remember from organic chemistry, it should become clear why this structure has partial double bond character and why it has restricted movement, and that's because of this double bond here. So as you will recall, double bonds can't freely rotate like single bonds can. Moving on, we're going to be talking about dipeptides, polypeptides, and proteins. And so the way I would think about these are really just definitions. So, your dipeptide is two amino acids, as seen here. Your tripeptide is three amino acids. Oligopeptides are anywhere from 2 to 20 amino acids, so they're usually considered smaller. And then polypeptides are anywhere from 20 to 50 amino acids. And so these are larger proteins. And then in general, a protein is, and this is the definition you should be able to have down for the MCAT, and a protein is one or more folded polypeptide chains with biological function. And so that last part is really important. Proteins need to have some sort of biological function. So they can't just be a polypeptide chain that, just floats around and does nothing. It needs to have some function, like they need to be enzymes or they need to be molecules like hemoglobin. It just has to be able to do something. All right. And then moving on, we're going to be talking about cysteine and disulfide bonds. So as you may recall from the previous video, this is the structure of cysteine. And the most important part of this structure in relation to disulfide bonds is the thiol group in its R group, which is this SH down here. And so what actually can happen sometimes is if pretend this long string is actually a series of amino acids without them drawn for simplicity. But and pretend that there's a cysteine here and a cysteine here, and these are the two thiol groups. It's possible that sometimes these thiol groups can actually interact with each other to make a disulfide bond, which is a linkage between them. And the reason this is so important is because these disulfide bonds are extremely strong and they are covalent bonds, unlike many other bonds within, a peptide. And so these disulfide bonds can stabilize tertiary structure within single polypeptides and even stabilize quaternary structure because of their strength. And so the way disulfide bonds are created is, again, you have two free thiol groups that are next to each other. And so this comes from one cysteine and one cysteine, as you see labeled in orange and labeled in blue. And then when these thiol groups interact to make a disulfide bond, this new molecule is actually called a cysteine. And so instead of cysteine with the E, it's just cysteine with an I. And that represents that, they're no longer two individual amino acids, now they're connected by a linkage. In addition to the formation of the disulfide bond, you're also going to see two hydrogens being expelled and two electrons. And so these are the hydrogens that are being lost and end up over here. And so because these hydrogens and more importantly the electrons are lost, this is an oxidation reaction. And so this reaction can be reversed through a reduction, but it is an oxidation when you go from two individual cysteines to a cysteine. And the reason this is an oxidation, as you may remember, is because you're losing electrons. And so an easy way I remember if a reaction is an oxidation or a reduction is just by remembering, the oil rig phase or phrase. So oxidation is loss of electrons. Reduction is gain. And as you can see, moving in the forward direction of this reaction, we are losing electrons because they're on the product side. All right, and finally, in today's lecture, we're just going to be talking about hydrolysis. So earlier we talked about peptide bond formation and how that is a condensation reaction because water is produced, but it's also possible to reverse that reaction. And so when it is reversed, we're going to be seeing a water molecule attacking that amide bond that was previously made, and then this is going to split it up into basically the components that you started with. So the oxygen from the water that's attacking is going to be seen in a free carboxyl group, and the waters and the hydrogens in the water that's attacking are going to be seen in a free amide group. And so this is exactly what we expect. If you release a water upon formation of a peptide bond, then it only makes sense that by using a water to destroy this bond, you get those components from the water back to where they initially started. And That's pretty much it for the lecture today. So it was, a very brief one, but it covers a few topics that are extremely high yield on the MCAT. So you're definitely going to see something about peptide bonds. It's highly likely that you'll see something about disulfide bridges, and so it's just good to have, a solid understanding of these concepts, what happens, and what they are.